Okay, I apologize if this looks like you're seeing more of the ceiling. I'm going to try and adjust it here. So there we go. Maybe that's a little better. I hope this doesn't make you dizzy. It's not quite flat, but it's better than if I try to do it onto the computer. And it's much better than if you have to look at me. So I'm thinking this is a winner. Okay, so last week we went over the granulocyte series maturation process or production process. And we're going to talk a little bit more this week about monocytes and lymphocytes. A little bit different, a little bit similar. We'll go through the similarities and the differences between the maturation process of the monocytes and the lymphocytes. One positive that we see is that we have fewer stages in the development of both the monocytes and the lymphocytes. So here we have in our bone marrow the production of monoblasts, promonocytes. When this cell gets released into the peripheral blood, we call it a monocyte. This cell also enters the tissues, and at that point we call it a macrophage. So we begin with the multipotential cell, the myeloid stem cell, or the colony-forming unit for the granulocytic monocytic series. This particular myeloid stem cell can differentiate either to a granulocyte or a monocytic series cell. When it differentiates to the colony-forming unit for the monocyte, we, in the end, get a monocyte or a macrophage. Same cell, just a different site. So again, this undergoes only 2 to 3 mitotic divisions from monoblast to promonocyte to monocyte. In a very similar process, lymphocytes mature from a lymphoblast to a prolymphocyte to B or T cells, which occur in the peripheral blood. Again, the production starts in the bone marrow compartments. We cannot differentiate between a T cell or a B cell in the peripheral blood. The B cells go on to produce plasma cells. So each of those lymphocytes function in a different manner, but come from the same production and actually look the same when we get into the peripheral blood. So both of these cells, again, now we're switching back a little bit. We're going to compare the similarities to the production of both these lines, monoblast and lymphoblast. The monoblast is an extremely large cell. You'll notice in these pictures that the nuclear to cytoplasmic ratio is about 4:1, meaning there's a lot of purple a lot of purple nucleus there. We typically see 1 to 2 nucleoli. You may see vacuoles in the blast. The key here, though, is that there are no granules produced in a monoblast. Very large cell, very soft and spongy looking, really, the blast cell is. The promonocyte is a monoblast which undergoes 2 to 3 mitotic divisions, and this division time takes 2 to 2.5 days, very similar in size, similar in nucleus to cytoplasmic ratio, a little bit smaller nucleus. You may see nucleoli, but you might not see any, so that in this case is not a differentiating factor. The cytoplasm is a blue-gray cytoplasm, so really it's just the amount of nucleus to cytoplasm that really will tell the difference, and it's very typical that you'd have to have both a myeloblast and a promonocyte to look at, so you could differentiate between the two. That mature monocyte then, a little bit smaller, but it is the largest mature cell that we're going to see in the bloodstream. Again, we start to see more cytoplasm, the nuclear to cytoplasm ratio 2:1 or 1:1. This nucleus is very mature. We don't see those soft, spongy nucleoli. We do see vacuoles occur. That cytoplasm, again, is a blue-gray color. They call it a ground glass appearance, kind of smudgy looking. And the nucleus, while very matured, can be folded or in a horseshoe shape. So sometimes this is the cell that gets confused with a granulocyte. You really want to compare cytoplasms for the presence of granules, for the dimension that's added by this ground glass appearance, and by the size of the cell. The monocyte is quite a bit bigger. So the monocyte, the mature monocyte, is released from the bone marrow into the blood between 12 and 24 hours after a promonocyte's last mitotic division. There are no large reserves of monocytes or promonocytes left in the bone marrow, but there are pools of monocytes and macrophages that are present throughout the body. So in the circulating pool, the half-life is only about 8.5 hours, so this cell is not going to last in the circulating blood system for a long period of time. It actually becomes too large to pass through the capillaries, so that's when it moves into the tissues and is converted into a macrophage. Here in the tissues, the macrophage will survive for months or even years. In addition to the circulating pool between the circulation and the tissues, there is a marginating pool. This marginating pool, which represents 3.5 times as many cells as the circulating pool, adhere to the vascular endothelium. So again, these are the cells that I would call the catfish. They're adhering to the endothelium and just taking debris out of its way. If we compare the sites of lymphocyte development, we have our primary lymphoid organs. These are sites of differentiation and maturation. And we have secondary lymphoid organs. The differing function here is that these are repositories or homes for already differentiated lymphocytes. So when you think of when you get sick with a virus and you have swollen lymph glands, these are found typically in the secondary lymphoid organs. So the primary site of lymphocyte development, again going through those 2 to 3 mitotic changes from a lymphoblast to a prolymphocyte, occur in the bone marrow for the B cell maturation, and this development occurs in the thymus for the T lymphocytes. The secondary lymphoid tissue, where the storage of these mature cells occurs, is primarily in our lymph nodes, in our spleen, and the Peyer's patches in the intestine. So our intestine truly is a part of our lymph system, which we're finding more and more interesting almost on a weekly basis now. So with our lymphocytes, these arise from a pluripotent stem cell. And the difference here, this differentiates into a colony-forming unit that's specifically lymphoid. So this differentiation is due to the influence of interleukin-1 and interleukin-6. When we start to discuss lymphocytes, the interleukins and measurements of their levels can give us some ideas of disease processes in the lymphocytes. So again, for both of the lymphocyte lineages are going to start with the lymphoblast stage. Again, we can't differentiate the differences in cells simply by looking at them. They next go into the prolymphocyte stage. Then they can turn either into a mature B cell where they transform into a plasma cell in the tissues, or they turn into a mature T cell, which is going to migrate then to the thymus to be stored. The lymphoblast, the first cell in the lymphocytic series in the bone marrow, nuclear to cytoplasmic ratio similar to the blast of the monocyte. It's about 4 to 1. You see a whole lot of big purple. You see 1 to 2 nucleoli, and you see no granules. So the difference here, if we look at differentiating lymphoblasts from monoblasts, is really we have very smooth staining cellular material. We don't have that ground glass appearance that we see with a monoblast. The promyelocyte, or the second stage of maturation, occurs either in the bone marrow or the thymus, or can also occur in the secondary lymphoid tissues. So we really look to that promyelocyte as the more mature. Anything past that blast stage is considered more of a mature lymphocyte. Similar nuclear to cytoplasmic ratio, you may see nucleoli, but you may not see nucleoli. And at this point, really, you rely on special stains to differentiate a promyelocyte. The B lymphocytes will multiply, and they'll live in these various secondary organs. They'll live in the follicular and medullary areas of the lymph nodes, in the primary follicles and red pulp of the spleen. They also appear in the follicular regions, gut-associated lymphoid tissue or GALT material, and we also find them in the medullary cords of lymph nodes. So the cords are a little bit more specific than what we see listed up with the follicular areas of the lymph nodes. The lymph nodes are filled with B lymphocytes. The secondary organ sites after the blast stage for T lymphocytes include the lymph nodes, again the parafollicular and paracortical regions of lymph nodes, so they're at more external parts of the lymph nodes. The medullary cords of the lymph nodes. Picture 59 in clinical hematology, you can check. I'm not sure that page is right, I have to apologize. I'll recheck and send you a note on that too. They're also found in the para-arterial regions of the spleen and the thoracic duct of the circulatory system. So worthwhile to take a break and go and see if you can find the specific sites of these cells. You can go back and look onto the dissection of the lymph node that occurs a couple slides previous to this and see what those different sites look like. Ah, there it is. So here we have a look at a lymph node. We have the afferent lymphatic vessel. We have the efferent lymphatic vessel. You can see the paracortex, the very light blue on the inside. The follicles are shown there. And when you think of your lymph node swelling, really the cells that are found in this follicular area contribute largely to that swelling that you see. So the differentiation between these B cells and T cells, they, as you remember, are categorized by immunologic function. So the connection to morphology can be made, and again, we don't see a differentiation when we look at the lymphocyte in the circulating blood. So the membrane characteristics of the B and the T cells, there are specific and special receptors on the cell membranes. For our B cells, there are membrane-bound antibody and complement receptors, because these, again, are the cells that are going to produce antibodies. And in a mature B lymphocyte, that specificity is to the specific surface immunoglobulins. On T cells, we have receptors to the CD cells that we use to identify and monitor cellular function in some viral diseases. So receptors for CD3, CD4, and CD8. So our B lymphocytes are responsible for our humoral or antibody response. They transform into plasma cells. The plasma cells are responsible for making the antibody to the foreign material once sensitized. And these cells aid in the defense of encapsulated bacteria. So our antibody response to foreign material, very heavily involved in bacterial infections. Take some time to look at the differences in the receptor sites on these cells, the names of the receptors, and again, remember the process of and production of the T cells versus the B cells, what the difference is between those two productions.